Hi, welcome. I'm Dr. Katy Martin, and I'm a member of the team here at miniPCR Bio. I'm excited to be hosting our webinar today, and I'm joined in the chat by a couple of my colleagues. If you have any questions as we go along, feel free to drop those in the chat and we'll get you taken care of. Today I'm going to be sharing our new Microbe Hunters Learning Lab with you. This is a dye-based electrophoresis lab that you'll be able to complete in a single class period. It is geared toward biotech beginners, but it's based on authentic space biology research. So we took some really sophisticated work happening out there in the real world and then kind of simplified it to make a lab that is accessible and engaging even to intro-level students. On this screen, I've snapshotted a few pages of the lab guide so you can see what I mean when I say this is targeted toward biotech beginners. Lots of information is presented visually and assessment questions are interspersed throughout to make this lab accessible even to students who aren't ready for really dense, text-heavy background information. These lab guides are available at minipcr.com. On the Microbe Hunters store page, which is linked in the description below this video, you can find all of the lab curriculum available to download for free. This includes the instructor's guide, the student's guide, and even PowerPoint slides that you can adapt for your classroom. Before we dive into today's lab, I want to start with an introduction. If you're new to miniPCR Bio, we make biotechnology tools and curriculum to make it easier for teachers to equip their students with modern laboratory tools. Our equipment is built to be accessible. That means more durable, more portable, and importantly, more affordable than typical lab equipment, so it can go places that ordinary lab equipment can't go. Among our users, we have scientists who bring our equipment to the Amazon to conduct field biology studies. We have hobbyists who use our equipment to identify the mushrooms growing in their own backyards. And we have epidemiologists who equip their pop-up labs with mini-PCRs so they can monitor emerging viruses. We even have equipment in space. This is a real, not a photoshopped image of a mini PCR floating in microgravity aboard the International Space Station. Since 2016, mini PCR equipment has been used aboard the ISS to do biology research in space. If you're not already familiar with it, the ISS is a science lab that is in continuous orbit around the Earth. It's been up there since the year 2000. It's been up there being built since 1998, continuously occupied since the year 2000 by crews of usually 6 or 7 astronauts at a time. These astronauts live and work in the habitable modules of the ISS, which are kind of hollow containers that make up the axis of the ISS. The most obvious thing you usually notice when you first look at the ISS are these big wings, and these are actually the solar panels that power the station. This stuff is not occupied, but the astronauts live in the tubes that make modules that make up the axis of the station. And those astronauts who live aboard the ISS, they're supported by much larger teams on the ground around the clock, including this one. This is the NASA microbiology team led by Dr. Sarah Wallace, you can see here at the left. Dr. Wallace and her team, they're based at NASA's Johnson Space Center in Houston, Texas, and they work to keep the astronauts safe from microbial pathogens, bacteria, fungi that can make you sick. And those are all the more dangerous to you when you're situated 250 vertical miles above the surface of the Earth. You lack immediate access to medical facilities. So this team's work was the direct inspiration for the lab that we're going to be doing today. In the Microbe Hunters Lab, the basic scenario is this. Your students are members of the NASA microbiology team. An astronaut aboard the ISS has just recovered from a dangerous bacterial infection. She was repairing a cargo hold with a screwdriver when her hand slipped, and she experienced a minor cut. It was nothing she couldn't patch up with a Band-Aid, so she just did just that, put a Band-Aid on it and went about her business. But over the coming days, she noticed that that cut began to grow red and inflamed. It started to ooze some pus. She called down to her doctors on Earth, and they advised that she apply antibiotic ointment to treat that infection. They were worried that if it was left untreated, it might spread into, say, her bloodstream where it could really wreak havoc. Now, fortunately, the astronaut has recovered, but her doctors are concerned that the pathogen that caused that infection is still lurking aboard the station where it might infect more crew members. So now a new crew is on its way to the station, and before they arrive, we need to make sure that the station is free from this dangerous microbe. The culprit, her doctors believe, is a type of bacteria. Now, of course, bacteria are single-celled organisms that live, capital E, everywhere, including all over the surfaces of our bodies, inside our mouths, throughout our digestive tracts, all throughout our respiratory system. While that might be kind of weird to think about, it's not a bad thing because most bacteria are harmless, and in fact, many help us out. But some can make us sick, so it can really pay to be aware of the bacteria that live among you. How do we look for bacteria? They are too small to see with the naked eye, so we can't simply look at a table and know what bacteria are growing on it. Instead, in our lab today, we're going to use DNA analysis to hunt for bacteria. And DNA, of course, is a molecule that can be found in virtually all cells. It contains the instructions cells need to do things like grow and divide, dispose of waste, really important functions. Those instructions are encoded in a sequence that we can read. And so those sequences are different for different types of bacteria, and we're going to take advantage of that here. It'll be like looking for the molecular fingerprints of the bacteria that we're interested in. Now, the name of the bacteria we're going to be looking for is Klebsiella aerogenes. We're going to call it Kleb for short. Kleb is the bacteria that are believed to have infected our astronaut. Kleb is actually very normal to find in the human digestive tract, but it can be harmful if it infects other parts of your body, say a cut on your skin or inside your lungs. We're not going to look for Kleb alone, though. We're also going to be looking for a second type of bacteria called Staphylococcus epidermidis, or staph for short. Now, staph is a bacterium that lives on human skin, so we're constantly leaving it behind on all the surfaces we touch. Staph is not dangerous to us, this type of staph. Looking for staph alongside our dangerous Kleb bacteria is going to serve a couple functions. First, it's more faithful to the way bacterial monitoring is usually done in ISS studies. Usually scientists are typically looking for many species at a time to get a more complete picture of the bacterial population on the ISS. Second, staph in this experiment is going to function a bit like a control for us. Since it can be such a common bacterium and it's found so widely throughout human habitats, we expect to see it in all of our samples. And if we don't, that's going to tip us off that something went wrong in our experimental process. It's going to serve as a bit of a sentinel for us today. Now, here's an overview of what this type of experiment would look like if it were being done in the real world. Hunting for bacteria involves a 3-step workflow. Now, Microbe Hunters really only encompasses the last step, but a lot had to happen before our experiment can begin. So it all starts with DNA collection. The astronauts swab, meaning they literally wipe a cotton swab across different ISS surfaces. If bacteria are present on those surfaces, a few of them will be picked up by those swabs. It's the very same principle that underlies microbial detection tests you might be more familiar with. So when you take a COVID test, for example, you start with a swab, right? It's the very same idea. In our experiment, those swabs were sent down to Earth and handed off to scientists at Johnson Space Center, where our team is located. Upon receiving those swabs, one of your teammates processed them with chemicals to extract the DNA from any bacteria or cells present on that sample. Another teammate then used a technique called PCR, or polymerase chain reaction, to copy segments of bacterial DNA. PCR is a very common laboratory technique. It basically allows you to ignore the noise, including all the DNA that we don't care about in that sample, DNA from other bacteria we're not looking for, DNA from human cells that might have been shed and picked up on that swab. We're going to ignore that noise and zoom in just on the DNA sequence that we care about. For us, that's going to be the sequences unique to Kleb and staph bacteria. Now, importantly, the length of the sequence that we copy with PCR is going to differ for Kleb and for staph. And then in the final step of our experiment, we're going to take advantage of that length difference to get our results. So this last step, this is where Microbe Hunters picks up. We're going to be using gel electrophoresis to actually see any of the DNA copies we made during the PCR step. If in a given sample we see DNA copies, that means the bacteria that correspond to that DNA must have been present on the swab that was used to generate that sample. If we don't see DNA copies for a given sample, that means we can infer those bacteria were not present on the swab. we're going to be able to tell those staph and Kleb bacteria apart in our gel because their DNA copies will differ in length. So let's talk for a minute about how we're able to use electrophoresis to separate DNA segments of different sizes. Electrophoresis is a way that we can separate molecules based on their size and charge. And if we break the word electrophoresis down, it's going to tell us how we're going to do that. So electro comes from electricity, and then phoresis comes from ancient Greek, and it means to be carried. So we'll use electricity to carry DNA through our gel and separate it based on size. At the end of the day, gel electrophoresis is a filtration process. When we're talking about microscopic scales and complex biomolecules, it can be kind of intimidating, but if your students can understand draining pasta in a colander, they can understand the science behind electrophoresis. Today, I'm going to be running electrophoresis in miniPCR's Bandit system. Bandit is a kit that students use to build their own electrophoresis system and then use it to run an experiment. We call this our build it to understand it approach. By building the system themselves, students have to engage with the scientific principles that underlie every gel run. This time lapse is showing you roughly how that's going to look. So this is time lapse into just a few seconds. But at the end of about a 20-minute run, we'll be able to see our results. Now, at the heart of our Bandit system is a gel made of a sugar called agarose. Under a microscope, you can see our agarose gel has a porous inner structure, a little bit like a sponge. We can represent that with this matrix of dots here for the sake of illustration. This gel is situated in the middle of an electric field. We can place a negative electrode at the top of our gel and a positive electrode at the bottom of our gel. And when we deposit our DNA samples in the gel, this electric field is going to force them to move. Since DNA is negatively charged, it's going to be drawn to the positive electrode. But you notice the DNA didn't migrate at the same rate. That's because the size of a DNA segment will impact how easily it can wiggle through the pores of our gel. Small segments will be able to kind of zip through those pores quickly, whereas large ones get hung up and don't move as far from the well in the same period of time. This is the basis of how electrophoresis separates DNA by size. Now let's zoom out and I'll show you how this will look to our unaided eye during our experiment. So here's our gel. We're going to load each of our DNA samples into its own pocket or well in our agarose gel. You'll see these here along the top. And any given sample might contain DNA segments of different sizes. When we turn on that electric field around our gel, our DNA segments are going to migrate through the gel toward the positive electrode along a straight path. We call the area under the well where the DNA will travel a lane. And as you can see, shorter segments will travel further than longer segments here. So segments of the same length remain in groups called bands. So each of these blue lines here is a different band. And one of our bands contains DNA segments all with shorter sizes. They were able to travel further from the gel and easily move through the pores. And then our second band, we know it consists of longer DNA pieces because they weren't able to travel as far from the well in the same period of time. Okay, so that's electrophoresis in a nutshell. I think we're ready to actually get on with our experiment and load our gel. All right, switching to my desktop cam so you can see my equipment. Here's my gel. I poured this before we started today. So what you're seeing now is Bandit in casting mode. I assembled all the parts to make a mold. Basically, I poured in molten agarose and that formed our gel. Now we're going to flip Bandit into run mode. So I'm going to start by pulling this comb. And each tooth of this comb creates one well. So you can see my wells here. Now these are dams that form the top and bottom edges of my gel. I'm going to take each of these dams and flip it now into run mode. And when I do that, you can see this electrode wire. It's going to be positioned right along the bottom of our electrophoresis chamber. And it's also going to create some space above the top of our gel and below the bottom of it. Here's my red dam now and my red electrode wire. Flipping this one as well. And creating that space is going to just help us when we need to assemble our electric circuit. Almost ready to load. First, I'm going to pour some buffer over this gel. This is TBE buffer. Buffer is just a conductive liquid that helps complete that electric circuit between our electrodes. Now we're ready to load our samples. So these are our samples. And I'm going to use a micropipette to load 10 microliters from each tube into its own well in our gel. And I'm going to be using a miniPET from miniPCR. Let me give you a good view of that. Our miniPETs are fixed volume micropipettes. So they're really nice for electrophoresis learners or biotechnology learners. You don't have to dial in the volume. So it's one less source of error, one less thing that can go wrong when you're learning. So these are really great training wheels. Now, the nice thing about these samples is they're pre-aliquoted. So the strip that you're looking at here, this is exactly what we ship you with the Microbe Hunters Lab. So it's really nice. You don't have to portion out samples into a million tiny tubes and hand those out to your students. It really reduces prep time where you really just hand one of these strips to each student group and they're ready to go. My third sample now. And I want to reiterate, these are dye samples. So they behave just like DNA, but they are just blue dyes. And the reason behind using those is you can visualize your results without any additional equipment. So as this gel runs, you'll see the bands moving. You don't need to transfer it to a different piece of equipment in order to see your bands. It just helps you get results faster and with less equipment. My fifth sample now. And loading gels, this is a lab for beginners. Loading gels can be really difficult for beginners. A day of micropipette practice before you have your students run this lab will pay dividends if they're brand new to biotech. It can make a huge difference between a successful gel run and one where you lose your sample or you poke your pipette tip through the edge of the gel and now you need to use up an extra well. Okay, we are all loaded up and ready to run. So we're going to connect our electrodes to our system. But which electrode is which? We want to make sure our electric circuit is oriented in the right way. This strip is like an indicator to tell students how they should clip their electrodes to the system. So this is how it works. If we clip our electrodes onto the positive and negative ends in this order, nothing happens. But if I take my red clip and clip it to the positive end of this light board, and my black clip to the negative end, there we go. Whoa. It tells us the direction of electrical flow, and DNA is going to obey that direction, right? DNA is negatively charged, just like electrons. And so that test strip tells us, okay, if you want your DNA to run through the gel, you want to clip your black electrode. Your black electrode is your negative electrode. We're going to clip that up above your well and clip the red one to the bottom. It's your positive electrode. And that is one of the biggest ways that a gel run can go wrong. If you clip your electrodes in the wrong orientation, your DNA samples pop out the top and you lose your samples. And so the light strip just helps reinforce kind of the science behind why we set it up this way to students. Okay, and I'm just wrapping this to get good contact. Great, I think we are set up and running. And in fact, I can tell this is working because I can see bubbles forming along my electrode wire here. That tells me this is working. Okay, let's let our samples run. I'm going to switch back to my slides now. Okay, so as our gel is running, let's take a second and talk about what those samples that we just loaded, what do they contain? Remember, the first step of our experiment is DNA collection. We asked the astronauts aboard the ISS to swab different surfaces, literally swipe them with cotton swabs, and those swabs will pick up any bacteria growing on that surface, among other things. Here's a photo of what that looks like when it happens in real life. So this is astronaut Ricky Arnold. He's aboard the ISS in this photo, and he is swabbing an area near the dining table aboard the ISS. You can tell it's the dining table because you can see hot sauce and ketchup here in front of him. A fun fact, too, about Ricky Arnold is he was actually a science teacher before he was recruited by NASA. So if any of you teachers in the audience have dreams of becoming an astronaut, there's still hope. You can still apply. It's happened before. So Ricky took some swabs, sent them back to Earth. The NASA microbiology team received those swabs and then analyzed them to see what kinds of bacteria were growing near that dining table on the ISS. They identified roughly 24 species of bacteria growing in just 10 square centimeters on the wall above that dining table. And this is Maybe not surprising because you can tell in this photo, this is actually a storage wall. Astronauts store their eating utensils here by Velcroing them to the wall. So this is a spoon that someone just used, licked clean, and then stuck on the wall. Maybe not surprising that there are tons of bacteria growing around that spoon. So where on the ISS did our samples come from for our experiment? So this is a floor plan. This shows you kind of the layout of the US portion of the International Space Station. And our first sample came from this module called Harmony. So modules are a little bit like rooms aboard the ISS. In Harmony, this is a space where some scientific research happens, and then up to 4 astronauts also have their sleeping quarters in Harmony module. In this photo, you can see 3 astronauts popping out of their respective sleeping chambers. Sleeping chambers are about the size of a porta potty, and they're kind of embedded in the wall. And in there, you'll find a sleeping bag that's kind of anchored to the wall. That's where the astronauts sleep, so they don't float freely during unconsciousness while they're sleeping. In our experiment, we asked the astronauts to sample the wall outside the Harmony module, so maybe here or here. Our second sample came from Destiny module. Destiny module is home to the ISS US National Laboratory, and this is a place where scientific research happens. We asked the astronauts to swab the laboratory countertop, the bench here in Destiny module, and we can imagine maybe this is a particularly clean surface, right? Keeping a clean bench is an important part of doing, good quality scientific research. But we can also imagine that, say one of our experiments involves culturing microbes in Petri dishes. Maybe one jumped out of its Petri dish and grew a little colony aboard the bench. We could see it kind of going either way. Our third sample came from Unity module. Unity is like the dining room of the ISS. This is where Ricky Arnold was sampling microbes a few slides ago. This time we asked the astronauts to actually swab the dining table itself. So this is a place where we definitely don't want dangerous bacteria growing. Our fourth sample came from Tranquility module. Tranquility is home to some exercise equipment, some storage areas, and also one of the bathrooms aboard the ISS. So what you're looking at in this picture, this is a space toilet. You can imagine this would be potentially a very interesting place to look for microbial life. We asked the astronauts to swab the handrail next to the toilet in Tranquility. So this is a surface you can imagine astronauts are touching a lot before and after they use the bathroom. Each of these samples was loaded into its own well in our electrophoresis gel. You can see how they lined up here. So we loaded our 4 samples into the 3rd through 6th wells. But what about those first 2 wells? Well, that's where we loaded 2 controls. So these controls are going to show us what it looks like when Staphylococcus or Klebsiella bacteria are present in a sample. We prepared these controls the same way we prepared our experimental samples, but rather than coming from ISS swabs, these samples originated with Kleb and Staph bacteria that we grew in a lab. So we are certain that these samples will contain the bacteria we're looking for. And unlike the rest of our samples, we can easily predict what we should see in these lanes. And in fact, these lanes are going to tell us exactly what it looks like in our gel when those bacteria are present in a sample. Now, our lab mate, before we moved on to the electrophoresis portion of this experiment, our lab mate used PCR to copy a DNA segment unique to each bacterium. And remember, they copied a long segment of our Staph bacteria, bacterial DNA, and a short segment of our Kleb DNA. And so when we run those samples in a gel, we should see that the Staph band is going to remain closer to the well. It's going to travel a shorter distance because it contains these long DNA segments that get hung up in the pores of our gel. Our Kleb band, in contrast, is going to travel further from the well in the same period of time. And that's because it contains these short DNA segments that have an easy time of moving through the pores of that gel. So at the end of the day, interpreting our ISS samples is just going to be a matter of looking at our experimental lanes and comparing those to what we see in our control lanes. Okay, our gel has a few minutes left to run. So while we wait, I'm going to tell you about another opportunity that we offer to engage your students in research and in space biology. Genes in Space is a contest that miniPCR runs in partnership with Boeing. We ask students in grades 7 through 12 in the US to design experiments that use biotechnology to address challenges faced by space travelers. And then each year we launch one winning experiment to the International Space Station where it is carried out by astronauts. Genes in Space pairs really well with Microbe Hunters. So if your students do Microbe Hunters and they're really excited about this experiment, about the ISS, Genes in Space is a great next step for them. It's a little bit more open-ended and can be driven, the direction it takes can be really driven by students' own interests. It also works the other way. So if you have students who are already really interested in Genes in Space, you're already planning on having them submit experimental proposals to our competition, Microbe Hunters is a great lab to start with. It can get them grounded in the context of space biology and introduce them to some of the tools that we use to address space biology problems. Genes in Space has been around since 2015. That year we named our first winner, the student Anna Sofia Bogorayev, you see up here. Anna Sofia was interested in studying the immune system of astronauts, and she developed a really interesting experiment to explore how the immune system changes during spaceflight. Her experiment required the use of PCR, this PCR technique that we've talked about in the Microbe Hunters Lab. And Anna Sofia's experiment was actually the first PCR experiment ever to be done in space that was carried out on the ISS when she was just a senior in high school. Now, since Anna Sofia, we have launched an additional 8 student experiments to space, and we are working on launching our 10th this summer. And each of these students has been responsible for their own advancement of the field of space biology. Each one is responsible for some first in the field of space biology. So it's a really exciting way to involve students in genuine, groundbreaking research. And we hope that your students are interested in helping us extend this timeline. In just a few months, we're going to be naming our 2023 winner. We're currently accepting applications. So if you know students who are great candidates for this, who are interested in space biology, in research, please send them our way. Genes in Space 2023 will be open again to students in grades 7 through 12. They can work individually or they can develop their proposals in pairs, and every proposal has to be sponsored by an adult. Often, that's going to be their science teacher. It could also be a parent or another mentor, as long as that person is over the age of 18. As their sponsor, you follow the student through the entire competition. If they win an award, part of their prize package goes to you. If they win the whole enchilada and they launch their research to space, you get to go with them to the rocket launch to watch the rocket go up. Submissions have to be sent to us by April 17th, and our application is available at genesinspace.org. Genes in Space is totally free to enter. There's no fee associated with participating. And I want to emphasize too, it's not like a science fair where you have to do a whole bunch of research and work and create a poster before you can go and enter the competition. Genes in Space is an ideas competition. So students come to us with their ideas, and then if we choose it as the top idea, we invest in them, we give them all the resources they need in order to get that experiment carried out. So the barrier to entry is as low as we could reasonably make it. Filling out our application requires about 1 page of writing, so a pretty reasonably sized science writing assignment. That application asks students for 4 things. We ask them what question they're trying to answer. We ask them what answer they think they're going to find to that question. What's their hypothesis? We ask them to justify their use of the ISS. The ISS is a very precious scientific resource. Many adult scientists who are adults and who have been doing research for decades want to do research on the ISS, don't have a venue to do it. So it's a really precious resource. We ask students to make a really great case for why they should be the one to use the ISS. And then we also ask them to tell us what their experimental plan is. We have a few items in our Genes in Space toolkit that have to be incorporated into each proposal. You can find more information about that toolkit on genesinspace.org, along with the application. So all of the stuff I'm talking about today can be found there on our website, along with many resources for both teachers and for students to help you get grounded and get the background information you need to craft really amazing proposals. So at this point, we're going to return to our experiment. So I just want to start with a little reminder of what we're going to be seeing. So remember, before we began, one of our teammates copied DNA segments from staph and club bacteria, a long DNA segment from our staph bacteria, a short DNA segment from our club bacteria. And so when we see long pieces of DNA in our gel, we know that's staph. When we see short pieces of DNA in our gel, we know that is club. This is what that's going to look like. So after our gel runs, we really don't know what we're going to see yet in our 4 experimental samples, but we do know what we're going to see in our 2 control lanes. And so what we're going to be doing in just a minute is comparing what we see in those experimental lanes to our controls to determine which bacteria were on which surface. Before we look at our gel, let's talk through a couple scenarios for what we might see and talk about how exactly we would interpret that. So let's simulate a gel run here and use this one lane for simplicity. Imagine in this ISS sample, you see a single band that runs roughly the same distance as the band you see in the staff control lane. This is what tells you that those samples contain the same DNA. So if it traveled the same distance, contains the same DNA, this is how we'd be able to tell if staff were present in a sample. The band distance from the well would match. Let's talk through another one. Okay, imagine we see 2 bands here in our ISS sample. We see one that traveled about the same distance as the band we see in our staff control lane, and another that traveled the same distance as we see in our club control lane. This is what it looks like when both club and staff are present in our sample. So bands, lanes can contain more than one band. Okay, let's talk through one more. What if we see nothing in one of our ISS samples? Well, that would mean neither club nor staff are present in that sample, but this is an unlikely outcome. Remember, staff lives on our skin, and so any surface that humans are regularly interacting with, including all of the surfaces we ask the astronauts to sample, they should have some staff on them. We would only expect to not see staff if we were sampling a sterile environment, and all of our sampling locations, we don't expect to have been sterile. Okay, I think we're ready. Let's go ahead and I'm going to switch back to my desktop cam so you can see my gel. Okay, here's our Bandit. And sure enough, we've had some movement. So the sample's no longer in the wells. They've moved down into the gel. And we see a bunch of different bands. So I'm going to go ahead and switch back now to my slides where I have a picture of what our results look like. The ones I'm seeing here in my system match what we've seen in the past. Okay, so this photo matches the gel I'm seeing here on my benchtop. This is from an earlier run of this experiment. And as we're talking through our results, let's first focus on our control lanes. So here, our staff control and our staff control here, our club control here. We can see one band in each lane. And sure enough, that staff band is very close to the well, meaning it contains large DNA segments. Club further from the well, that means it contains smaller DNA segments. This matches what we expect to see based on the design of our PCR experiment. So great. Control lanes look as expected. That's good. What about our experimental lanes, our ISS samples? Well, the first thing we might notice is that there is one band that we can see in all 4 of our experimental samples. This band has traveled the same distance from the well as the band we see in the staff lane. And so that means that these contain the same DNA. We can see staff DNA in all 4 of our ISS samples. And again, that's not surprising, right? We know staff lives on our skin. We leave it behind everywhere. So it's actually a good thing that we see it in all of our experimental lanes. That means our experiment's behaving as we expect it to. But what about our Kleb bacteria? So we do see 2 bands in 2 of our ISS samples. And that band is the same distance from the well as the band we see in our Kleb control lane. And so, oh boy, that means we have Kleb bacteria lurking in the Harmony module, in the Tranquility module. Yikes, we need to get that out of there before our new crew of astronauts arrives. So the outcome of this lab is, hey, we need to radio up to the astronauts. We need to tell them, get Harmony and Tranquility on your housekeeping chore list for this weekend because they need to be disinfected and deep cleaned to eradicate those Kleb bacteria before the new crew arrives and is potentially infected. So this is where our lab ends, but it's where some really interesting conversations can begin with your students. So you might ask students, how do you think CLEB got into these modules in the first place? Tranquility in particular, remember that's where our space toilet's located. It doesn't take much to connect the dots and say, okay, if we have a bacterium that grows commonly in our gut and we find it in the module where the toilet is, what do you think might have happened to kind of bridge that gap? You can also talk about the practicalities of space research. So why would NASA do these monitoring studies instead of, say, just deep cleaning the entire ISS by default before a new crew arrives? What are the practical impediments that might make that not the typical way that we run the cleaning program on the ISS? So some really interesting conversations can be seeded by this lab. And to wrap us up today, I'm going to just reinforce this is a really engaging scenario-based lab based on real ISS research. Again, it's written for biotech beginners, so it's a really accessible on-ramp into biotechnology for students as young as middle school. It can be an interdisciplinary introduction to biotech as well if you use the Bandit electrophoresis system. That allows you to cover some of the physical science principles that underlie electrophoresis while you're also talking about the life science problems and challenges that can be addressed with that technology. And as we did today, you can get your results in one class period. So you don't need to worry about managing equipment over multiple days. Bring out your bandits, run your experiment, put them away. You can do this entire workflow in just 45 minutes. I want to remind you that all of this curriculum that we went through today is accessible for free on the store page on the minipcr.com website. You can find the lab guides for the protocol we just completed, as well as some PowerPoint slides that you can use in your classroom. And if you were to order the Microbe Hunters Lab Kit, that comes with the pre-aliquoted samples that we ran today. So up to 8 groups of students can do this lab. So we send you 8 of these sample strips that are all ready to load. We also send you the reagents you need to make gels. So agarose tabs, these contain pre-measured agarose. That's just add TBE buffer and you'll be ready to pour your gels. We send you the TBE buffer you need in a single serving pouch. So we make it as easy in terms of prep as we possibly could. The hardware that we used to run this lab was our Bandit STEM Electrophoresis Kit. This lab can be run on any electrophoresis equipment that you might already have, but if you don't already do electrophoresis with your students and you are looking for systems that you can use, Bandit is one of the most accessible ways to run electrophoresis. Accessible both in terms of learning, it really helps students understand the science of electrophoresis. Also accessible in terms of price. You can buy one Bandit system for under $100, and you can also buy it in bundles of 4 or 8 bandits along with some other accessory equipment and equip your entire class for under $500. If you're really interested in trying out Bandit for yourself, we actually hold virtual workshops where we send you an equipment kit, including a Bandit, and then we get together on a Zoom to go through the Microbe Hunters experiment. So you'll receive all of the equipment and reagents you need to do Microbe Hunters together with the miniPCR team. We still have space available in the session that we're holding on Saturday, April 1st. So if you're interested, we encourage you to email outreach@minipcr.com and they'll send you details on how to register. There is a cost associated with these virtual workshops, but it's only $25 and you'll receive about $100 worth of equipment and reagents. So it's a really good deal and a great way to try out Bandit if you're interested in what I've shown you today. miniPCR, we also have other materials for biotech beginners to get them on the road toward learning biology and biotechnology. We have other dielectrophoresis labs that were kind of built to use with the Bandit system. We have a cat genetics lab, which is a really nice exploration of a Mendelian trait in a family of cats. And we also have a lab called Molecular Rainbow, which is a really great way to talk about the properties of molecules and observe how those affect the way they run on electrophoresis gel. We also have a new lab that we just released called Full Steam Ahead. This is actually a way to train your students in micropipetting using micropipette art, a really fun, easy, low-stakes way to learn micropipetting. It's really scary for students to load a gel the first time they ever micropipette. They just feel so much, it can be such a worry if they don't get it exactly right. They want to screw up their experiment for their group. But micropipette art is like paint by number. It's fun, it's relaxing, and it's an easy, low-stakes way to kind of build that muscle memory to ensure proficient pipetting down the road. All right, that wraps up our webinar today. I want to thank you guys for joining me. And again, if you have any questions, please feel free to drop those in the chat. We'll hang around and address them. But thanks so much for joining me. Bye-bye.